Thank you all for coming today. It's a Sunday and a gloomy Sunday in Ankara. And instead of staying at your homes in bed watching Netflix, which I would have done if I wasn't here. So it's a very good thing being here. Congratulations to you as an audience. And I want to congratulate all the organizers. It's an amazing job. And I've been to, as you may guess, to many conferences organized by companies like specialized in organizing congresses. And you are much more professional than them. So congratulations, amazing job. And if you want a reference letter on this, you're welcome. I can give clips. And for the people whose emails I haven't responded, I'm so sorry. We are popular. We receive a lot of emails. Not because of that. Nobody is busy, too busy to respond an email, but because I want to make a disclaimer on this. If the answer is a definite yes, which is impossible because we are full, and unless it's a definite no, we cannot give a reply to emails because I can't say yes because we don't have space. I can't say no because it's a good CV. So your email just floats like that and unanswered. So I'm sorry for that. So modeling Alzheimer's disease in zebrafish. I will take this. First of all, my group. This is my group. Most of you know Umutcan, previous president of the club. So he's in my group. This is me. Elif and Ceren is graduated from METÜ, Molecular Biyoloji and Genetics. And Tuba Sena is from Boğaziçi. Elif, Sena, Ceylin, Reyhan, and Umutcan are from Bilkent, and Ali Eren is the only non-MBG person. He's from Top University. He's a material person. So this is our group, graduates, and we have a couple of undergraduates around. Some of them are here. Our lab is Arslan Ergül Lab. We finished our third year this November, so it's 3 years old. And we are based in UNAM. If you don't know UNAM, you don't. And that's normal because we don't enroll undergraduates. Like it's only a graduate program, material science and nanotechnology. It might seem to you ununderstandable what is material science and nanotechnology, but it's an umbrella term. So there are lots of physicists and engineering people and people from all over the areas in sciences that are doing science together because science has to be interdisciplinary and collaborative. So UNAM is a research center. It is located between Science A and Science B buildings in Bilkent University here in the campus. And it's 6 floors and 6 floors all laboratories. It's a whole research center. It's a perfect place if you want to do science only, which is me. So it's a perfect place for me. We don't have undergraduate program. You only enroll through graduate programs, which opens 3 times in a year. We have 3 enrollment times. Please check the website and don't be intimidated by the words like nanotechnology and stuff. Just look at the people working in UNAM and see what they're doing and apply for our programs. The next one is coming in March, I think, but we have all year-round applications. So feel free to look at them. And do not be discouraged by emails unanswered because, as I explained before, just take your chances. The only critical thing in our applications is that we need an IELTS or TOEFL score. So make sure you have one, and also a GPA 2.8. Other than that, all depends on if you want to make science or not. So we don't have restrictions on undergraduate degree area or what you are doing or where you are from. We have no restrictions. So what we do as an Arslan Ergül Lab, first of all, we are an outreach group. I say first of all because outreach comes before our research activities. We take pride and we emphasize this a lot. First of all, we go to high schools and high schools come to us. I go to high schools in this Tuesday, I have another high school visit. High schools invite me, I go there and I talk and I talk and I talk. I can talk hours and hours long so that they can see what a scientist looks like. And if they have any questions about being a scientist, they can ask. They're usually the same questions. I'm doing this for almost 10 years now. And also I have a YouTube channel, Ayça Ergül. How conveniently named, that's my name. So I'm posting on YouTube all the time, Turkish, English, book reviews, courses, everything. I was teaching in Moleküler Biyoloji and Genetics department in Bilkent University for years. And then I said, I'm so good at it, why not give it to the whole world? So I transferred it to YouTube. And now UNAM offered me a position where I won't teach in a classroom, but on YouTube as well. So it's part of my job. It's not bedava. It's my job, by the way. So it's serious. Like it's not a hobby. I have many hobbies, but this is not a hobby. It's job. It's serious. So follow me, subscribe me, like me, whatever, and on YouTube. I don't see your phones. Just take them out and subscribe my channel immediately. So as a research, we do zebrafish, C. elegans, and human cells. I know nothing about C. elegans. That's because Umutcan wanted to work on C. elegans, and he's working, and I have no idea what they're doing. And there is Elif Sena and Maria. Maria, you know, from MBG department. She's also working on C. elegans. It's a funny organism. It has like numbers of, like quantifiable numbers of neurons. Like, for example, if you want to work on dopaminergic neurons, for example, in humans, how many dopaminergic neurons do we have? Lots of. But in C. elegans, there's only 8. So if there is 9 instead of 8, it means there's an increase in dopaminergic pathway. It's that simple. So we like simplicity of the animal and we work on it. You wonder how it is translatable to humans, like an organism having 8 neurons, dopaminergic neurons, but it is relatable. And C. elegans studies has contributed to science a lot. For example, now everybody knows that caloric restriction is beneficial for aging, right? Everybody knows that. We don't apply that rule, but we know. So it came from C. elegans, for example. It is found in C. elegans. So that's an important animal. But the most important animal on Earth is zebrafish, obviously. Zebrafish is a wonderful species. We like them a lot. We cut them and take their brains out, but we like them eventually. Disclaimer, we do everything under anesthesia. Like even in fish, we give anesthesia first and then we poke the fish. So if it is responding, if it's not responding, then we cut the brains out. So that's all humane work. And human cells, not interesting, so I skip that part. So we have some important research going on. First one is a very important research, AD Imprint. It's a European Union joint program on neurodegenerative disease research. We have collaborators from Germany, Switzerland, Netherlands, and again, Germany. because Germans are everywhere, and we are here. This is just started. So this is about Alzheimer's, but I won't give you any results on this project, not because it's highly confidential and important and super intelligent, but because we don't have any results yet. It's just started. And also on Parkinson's, again, the same program, but the people are from Denmark, Germany, Sweden, and Norway. So all Scandinavian and us as non-Scandinavian. So it's very good to be in modern world people together making science. So in these both projects, we use zebrafish as a model, not C. elegans. C. elegans people fail to get any European Union project, but zebrafish people do. So that's a competition and a message to people among us to do something. Okay. So we both use zebrafish as a model in this. And also we have some TÜSEP projects. TÜSEP is the Science Institute, Health Institute in Turkey. And here we see a small project on C. elegans. Not enough people, not enough, more money. I need more money. These are small projects. So I skip them. So let's come to the science part, Alzheimer's disease. Alzheimer's disease used to be very simple in teaching 10 years ago. 10 years ago, when I was teaching like Alzheimer's in a class, I would say that there's the amyloid and it goes wrong and it develops in the brain and then you get Alzheimer's. But now things got complicated, too complicated that I don't know if anybody can solve the problem right now because it's more than amyloid. But in as a like person view, my grandmother died of Alzheimer's, by the way. God bless her. She was old, so you don't need to be sorry right now. So she got Alzheimer's, and it is defined by memory loss, but a different type of memory loss, you may know. You start losing your memory from to backward, like you remember your childhood, but you lost contact with a year ago, for example. So it's a different type of memory loss. So it's not dementia, by the way, it's different. But dementia is also present sometimes. But among that, the patients lose reasoning and language, like forming complete sentences, coordination, like tying your laces, and mood swings and behavior. Nobody is interested if an Alzheimer patient is depressed. So because she's already Alzheimer's, how worse it can get. But they get depression, anxiety, and mood swings, and so on. And Alzheimer's disease is also hard, difficult, because it's never alone. Because in Alzheimer's patients, because they're aged, old people, it comes with other diseases. So in here, the what is this? Orange. Orange color is autopsy confirmed AD. What do we mean by autopsy confirmed AD? Because you cannot take a sample from brain, or since there is no definitive diagnosis of Alzheimer's disease, if you want to be 100% Alzheimer diagnosis, you have to do it post-mortem when the patient is dead. So after that, after autopsy, they can say that it is 100% Alzheimer's. And it comes with hippocampal sclerosis. Sclerosis is, you listened to multiple sclerosis today. So hippocampal sclerosis is the death of neurons or dying or degeneration in the hippocampus. It's a disease. And cerebrovascular disease, this is heart problems. And also frontotemporal lumbar dementia and Lewy body dementia. These are all different diseases. So it is becoming complicated, as you can see. It's not just Alzheimer's with dementia. There are lots of other diseases. And these are different from Alzheimer's. So a person can have both hippocampal sclerosis and Alzheimer's and frontotemporal lumbar dementia together. So you have to treat them differently. The other thing is genetics is complicated in Alzheimer's. This is the patients, each of these dots is a patient or finding or a sample, and each one is a mutation. So you can see how many different mutations are present in Alzheimer's disease, and some are very significant, like here TREM2 is very significant, or ApoE is very significant, but you cannot tell that this gene is responsible from Alzheimer's. So it closes a chapter, so you can close the chapter on finding the genetics on Alzheimer's disease, because there's no one single responsible gene. Why it is important? It is important because if it were like one gene, you could have knocked it down, and then you will have an Alzheimer model in animals. But now you can't do that. What is, for example, SMA, spinous atrophy. It has only one gene responsible from it, SMN. And if you replace the SMN gene product, for example, you can have a therapy, which is nusinersen or Zolgensma available. But in Alzheimer's, it's not possible. So we skip the genetics part because of that. And when we come to pathology, the amyloid hypothesis is well known. There was some discussions about amyloid papers, and amyloid is not being responsible from Alzheimer's. You may have heard that. Amyloid is still responsible from Alzheimer's, but not 100%. Again, not one pathology is responsible from Alzheimer's. That's another complication. But if you look at the amyloid, so amyloid precursor protein is this, and there is an enzyme here, secretase alpha. It cuts this protein into two, and then you have two portions. This is the C-terminal fragment, and this is the extracellular fragment. And then there is another enzyme, gamma secretase, comes and cleaves this part further, and then you have two peptides. One is inside the cell here, and one is outside the cell. So this is what happens normally. One important thing here, amyloid per se is not something dangerous, like cholesterol. People always say that cholesterol is toxic, cholesterol is this bad, cholesterol is that bad, but you cannot live without cholesterol. Cholesterol is an essential part of cellular membranes. So what is dangerous is that you have extra cholesterol molecules running in your bloodstream. So that's the dangerous part. Like sugar, for example, sugar is not toxic per se, but if sugar levels in your bloodstream is high, then it is dangerous. So in case of amyloid, it's a very beneficial, very nice, easygoing protein in our neurons and some other cells. But what happens is that it goes wrong at some point. Like here, this is called the amyloidogenic pathway. Now another enzyme comes here, beta secretase. It cuts at a different position. Then gamma secretase cuts this. Now we have two peptides. This one is same inside the cell, but the one outside the cell, extracellular domain, is amyloid beta. And this is the dangerous peptide. Why it is dangerous? Because amyloid beta peptide forms aggregates. What is aggregates? Aggregates is like they stick to each other some way, and when they stick to each other, they become non-functional. And for a protein, it has to be functional. So in the regular lifetime of a protein, of a normal protein, it is synthesized from mRNA, and then it does some function, goes and cuts something and do some protein things in the cell. And then proteasome comes and degrades that protein or peptide into small fragments like amino acids so that it can be recycled. Nothing lives forever in a cell. It has to be recycled and remade. But if it's an aggregate, like they stick together, not only they're non-functional, but the proteasome cannot degrade them and cannot recycle them. And they stay as a unused item in a cell. And if a person is 80 years old, it means that for 80 years, they become more and more aggregates in the cell at a point where the cell cannot function properly. And if that cell is responsible for keeping your memories intact, then you will have memory problems. Or if that cell is in your motor area in the brain, then it means that your motor functions cannot do its job properly. So that's what aggregate means. But as I said, things have become nasty and complicated, as I said. Now, amyloid is not enough. It is necessary. In every AD patient, there is amyloid plaques, amyloid aggregates, but amyloid aggregates are not enough for Alzheimer's. So there are some meditating people in Tibet, in a monastery. These are like 1,000 years old people. So they die and they donate their brains to science. And when they do the autopsy, there are lots of amyloid plaques in their brains, but they were not Alzheimer's. So amyloid is not enough. So we need something else. That something else is tau. Tau is another protein. It also has the function or ability to form aggregates. It's normally a very useful protein, but it can form aggregates. But in this situation, we call them tau fibrils because just to make your life complicated. We call amyloid plaques and tau fibrils. So tau make fibrils. Where is it? Here. They form fibrils. And they also prevent the function of the cell for reasons we don't 100% sure. So in here we have homeostatic microglia and activated microglia. In here there is a line. This part is preclinical AD. It is the Alzheimer's disease, but still you don't see Alzheimer's. So here another important thing, Alzheimer's disease actually starts 10 or 20 years before the first symptoms arrive. So when you see someone who is having problems remembering what they did 1 week ago, it means that the disease has started 20 years ago. So that's a very nasty thing because I am 45, I might have started developing Alzheimer's and there's no way that I know about that. So that's scary. But we won't dwell on that thought. Now here is preclinical AD. We have microglia and astrocytes. In brain, we have neurons, as you know, the main cells in the brain, the neurons, but there are also assisting cells to the neurons, which are called glia as a general, but there are some subdivisions. Astrocytes are like the repair, damage and repair and maintenance guys in the brain, and microglia are the immune system. So our normal immune system, the T cells, B cells, and so on, they do not go up into the brain. Brain has its own immune system, which is called microglia. So the amyloid hypothesis is the first one. Tau hypothesis is the second one. And the third one is neuroinflammation. Neuroinflammation means that the microglia and astrocytes become activated in the brain. And we don't like that. We don't like activated immune system. When the normal immune system is activated, you start sneezing and coughing and have a fever, for example. But if the immune system in your brain gets activated, it means your brain is in fire and cannot function properly. So you know that immune system is very drama queen. Normally, even if there is a small virus, it becomes so much thing out of it, like a fever or coughing, and you stay in bed. It's not because of the virus, but because of the immune system. The same is true for the brain. If the brain's immune system is activated, it starts meddling with your brain, like brain fog, as we've seen in COVID, or memory problems, or movement problems, or behavior problems, for example. So that's three items we need to think about. And if you look at the normal neuron, in normal interaction of a neuron and an astrocyte, so here there is the presynaptic neuron which sends the signal, postsynaptic neuron which receives the signal, and there is the astrocyte which is the helper guy here. So there is the neurotransmitters and some receptors here. Neurotransmitters open and close these receptors, and the receptors pass through, enable the passing of sodium, calcium, potassium, and so on. So it sends a signal, and this is how your brain works, basically. But in neurodegeneration, first there is a microglial activation. So you see there are amyloid plaques and the tau fibrils, and these call in for microglia, and microglia makes a fuss around here. And then there is astrocytes taking too much of the neurotransmitters in here, more than they should. And then also there is this degeneration going on here because these misfolded and aggregated proteins interfere with the release of the neurotransmitters. So they do not either release any neurotransmitters, or they release too much of neurotransmitters. And we have postsynaptic density here, which is the essential for receiving the signals. So the misfolded proteins can interact with this area as well. So the whole neuronal transmission is somehow affected. And there are some therapy options. Currently, there is no effective therapy for Alzheimer's, maybe you know, but there are some strategies like strengthening the synapses, protecting synapses from toxic stimuli, and preventing degeneration. And here also, there is a nice diagram. The blue you see here is pyramidal excitatory neuron. This is an excitatory neuron. Excitatory neuron is like, it's not like makes you happy or so excited, but it just sends signals instead of blocking something. So I move my arm like this. It's because of the excitatory signaling. But if I can't stop moving my arm like this, for example, it will be too much excitatory signaling, so on. So in order for me to stop my arm swinging like this, we need GABAergic inhibitory neurons. GABAergic inhibitory neurons just say it's enough, honey, just stay calm and do not swing your arm signal here. And also we have microglia and astrocytes helping things around. So the therapy options, the paper 2023, Nature Review Drug Discovery, it says that therapeutic options for Alzheimer's, do not bother reading it. It's 23 pages or so. It doesn't tell you anything. So I read for you, and as a summary, there is no therapy yet. That's the summary. Of course, it is 4 pages long tables listing lots of chemicals interfering with all of these, but so result, is there a drug I can use? No. Keep working, people, keep working. So we come to our magical, amazing, unbelievably wonderful organism, zebrafish. When you see it in here, you may think that it's a big fish. It's not. It's only at its largest, it's only 4 centimeters long. It's very small. Why do we work on zebrafish? Because it is cheap. It doesn't require too much funding, which we don't have. And it requires only a little bit of space, which we have only a little bit of space. Those are the reasons that we work on zebrafish. But the scientific reasons are the ones we tell in each presentation is rapid development and external development. What does it mean? It doesn't develop in the womb. The male just poofs the sperms, and the female just poofs the eggs. If they don't poof, I make them poof by squeezing. And then it is external fertilization. And then they develop on their own. And you have to separate them from the mother because mother eats them back. So it's a very empathetic. It has feelings, you know, but cannot differentiate its own child from the food. Yeah, it doesn't differentiate food from the child. So so it's a problem, but we solve it, and you get lots of progeny. What is the importance of this rapid and external development? You can we have 1 hour period while the fish is fertilized egg is only 1 cell. So 1 cell is very important if you want to make transgenic animal, because you have to change the genetic material while it is only at 1 cell stage. If it is 2 cell stage, you've missed your period chance of doing a transgenic, because then you will have half of the cells transgenic and half of the cells normal in the organism. So as a single cell, you have to inject whatever you want to inject. So it is important. Transparent, for the first 7 days, it is fully transparent. You can see all the organs developing inside, which means you don't have to cut and dissect anything. You can just put the whole animal under the microscope and see what is going on. If you want to observe the liver formation, you can just see it by looking at it. So it's that simple. And high throughput screening, because it has lots of children, hundreds of each time. Genetic malleability, you can change the genetics, the whole genome. Transparency, as you can see through. Cost and time efficient, most important thing. And molecular and genetics homologs, 87%. Our genome is 70% identical with this guy. So evolution, guys, evolution. 70%. Like you are, as a human being, a highly important, highly complicated being, and still your 70% of your genes are exactly the same. By meaning exactly the same, I say GTC, like sequence is exactly the same with this cute little thing. And it's a vertebrate, very important. C. elegans is not a vertebrate, guys. Zebrafish is vertebrate. So do you remember the animal kingdom, and then it goes on top, very top, there's the vertebrates. So it is from there connecting with the human. And these are the all cute organs. Gills, we don't have them, but heart, intestine, spleen, pancreas, urogenitals, testes, not remember really necessary, muscles, and kidney. So these are the common organs with human, which means that you can work on any of these organs. If you have a kidney disease to make a model, so you can use zebrafish. It means that only lung, you can't use lung, it doesn't have any lung. It is important, you know. And also, when we come to the brain, so this is the human brain, telencephalon, diencephalon, olfactory bulb, spinal cord, cerebellum, and zebrafish have them all. It has a huge olfactory bulb in relation to other parts because olfaction is really important for zebrafish, even though it doesn't differentiate its child. But telencephalon is very important because telencephalon is this huge part in the human brain, and it is very small in zebrafish, which is very convenient to work on, and also the spinal cord, and also cerebellum. So you can make models of each of these brain parts in zebrafish. These are the pictures from our lab. Reyyan, our master student, took them. So it starts, this is the one cell, but the huge thing in here is not the cell itself. The cell is coming from here. This is the yolk, you know, yolk, the food providing, and two cells here. So you need to inject in this stage, and then it divides, you know, bubbles, more bubbles, lots of cells, and then at the end of 24 hours, you got this larva. You got this larva at the end of 24 or 72. I'm not sure, because I'm not the one who spent my 72 hours in front of this microscope. It was Reyyan, obviously. As a PI, I don't do those mundane works. I just tell them, take the pictures every hour and then go. So that's the beauty of my life. So Reyyan took these pictures, and also she took this. This is the actual picture of 1 centimeter. This is the adult, like 6 months, I think, or 12 months. This is bigger because it has eggs in here. It looks like pregnant, but it is unfertilized eggs in here, abdomen. And this is the tiny larva. So it looks like this. And what you can do with zebrafish, first of all, behavior tests. You can do behavior. Behavior is very hard to do in humans because you cannot control all the external environment. So there was this caloric restriction study in humans. It failed. Do you know why it failed? Because they didn't obey the diet. Because you cannot put people under observation all the time and watch what they're eating. It will be too stressful. So they sent them home. After 6 months, they ate whatever they like. So it is easier in zebrafish. You can do lots of learning tests. These are learning tests, by the way. And also you can do lots of motor response tests, like vision and like startle response. Stigmotaxis is like you just poke them. And then again, around vision, prey capture is my favorite. You just put some tool that we catch the fish. So you just put it and chase the fish. And they are stressed, obviously. I like it. T maze and Y maze, they can learn. By the way, they can be little fishes that, and they can be so small and insignificant to you. They are extreme behavioral development. First of all, you cannot take one fish if it is old, for example, 12-month-old, from its tank and put it into another tank. You cannot do that. It's their home, and they know their home. And if you put them in another tank, they will be extremely stressed. And you cannot put them in isolation. You have to be in communities. They have communities, and they have alpha beta system, like alpha male, beta male system, really do, unfortunately. And they get depressed, they get anxious, they get nervous. I induced all of them to the poor fish, I know. When they are depressed, they just stay at the very bottom of the tank, unresponsive. So very similar to human. That's good. And also in, this is the very similar picture, but we usually take the telencephalon and cut it into pieces. And then you can see what is going on in their brain at the cellular level. Because as you may remember, we have 70% identical genomes. So if you do something like if you put it into depression and then euthanize it and then cut the brain in slices, you can see the cellular mechanisms, which is different in the fish brain, which is impossible to make in humans. So that's the good thing. And also it is kind of impossible in doing it in mouse. I won't go into details of mouse, but this is complicated in mouse. So it is more simpler in zebrafish. And what we do direct micro injections into the brain. We can do that. And we can do injections into abdomen or heart so that we can induce something. So we can do transgenic fish. As I said, in the one cell stage, we can change the genome. But you remember, Alzheimer is not a single gene disease. So you cannot make a model of Alzheimer's disease genetically. You have to induce it by giving the clumps or aggregates of the protein. So the zebrafish itself, you cannot ask zebrafish if it remembers its childhood memories, but you can give fibrils of tau or fibrils of amyloids or plaques of amyloids into the brain and then see what happens at the cellular level. So as I said in the beginning, we don't exactly know what happens, how amyloid plaques inhibit something. And in our own project, we are employing a zebrafish. We didn't do it, we just got it from Belgium. That's another good thing about zebrafish. So you find someone working with a model and you ask them the embryos and then they bring you the embryos. That's that simple, easy, free, without charge. It's a gift. So we got from Belgium, a graduate from University was working. So we emailed him. He said, oh, sure, I can bring it while I'm coming to Turkey. And he came to Turkey to visit his parents and brought us the transgenic fish, which is the MPEG M-cherry fish, which means MPEG is a marker, biomarker of microglia. It's a promoter. So you take the promoter of MPEG and then instead of MPEG protein, you put M-cherry, which is a fluorescent protein. So now our fish has pink microglia. Literally, it's under fluorescence. It glows as pink, which we prefer instead of GFP. Pink is much nicer. And also we have GFAP fish. GFAP is the biomarker of astrocytes. And GFP is in green. So when you mix them, what we get, hopefully, we haven't confirmed yet, we just hope, that the astrocytes will be in green and the microglia will be in pink, and then we will inject the amyloid fibrils, and then we will see what happens in those cells. So you have to know when you cut the cell, brain into slices, you need to know which cell is which. You can't identify it later. So if it is pink, we will say that it is microglia, and we will see if the amyloid is accumulated in microglia or not, and we can see things like that. And it will look something like this. This is from Çağhan's lab, Çağhan Kızıl. Çağhan Kızıl, you know, he's graduated from ODTÜ, MBG. He's very good. Just Google him, Çağhan Kızıl. He's good. And he's in Colombia right now. That will affect you now, your search. He's in Columbia University. So amyloid beta is in green. This is a section of the zebrafish brain. Conveniently looks like a heart shape. How lovely it is. But it is a section from telencephalon. And DAPI shows the cells, neuronal bodies, the DNA, and the amyloid beta fibrils are in green. So you can see such a picture. And if you combine it with GFP, like astrocyte microglia, you can know which cell it is. And there are some drawbacks, obviously. So in humans, we have neuronal death and cognitive deficits. But in fish, no cognitive test assays are available. Like you can test learning, but it will be new learning. As I said, it won't be like childhood amnesia type of test. So it's a drawback. And we will have increased numbers of activated microglia, which is same. That's a good thing for zebrafish. And we are focusing on microglia anyway. And you see this also, but you have to give human version. Like zebrafish doesn't normally develop amyloid plaques, as you might guess. They don't live 80 years. In humans, it develops in 80 years in a cell. Zebrafish doesn't live that long, or we don't have that much time. So we just inject the human proteins, and amyloid beta plaques also form. So it's good, mostly beneficial. So I'm tired. Do you have questions? This is my lab page. Go check it out. Unfortunately, we don't have places for new interns, so but send an email anyway. I'm ashamed to say this because we receive too much applications, which is a good thing for us, but bragging about, but we don't have too much spaces. Really, we don't have too much spaces, meaning physical spaces. The lab is so small, we cannot fit into the space available. That's the reason. So if we have larger laboratory, then we will let you know. Thank you for listening.
Okay, so we have still some time for Q&A. If you have any questions, please raise a hand. There's a question there.
Okay. Can lots of aggregates can be degraded? Can amyloid do the same with some protease? So we can, potentially decrease the effect of Alzheimer's.
I think there is a therapy option exactly like that they're working on it. But I can see the most initial problem would be like it will degrade other proteins as well, because the proteases are not that specific. The kinases are more specific, I think, but they're working on it. I'm sure it was on the list.
Okay, thank you.
Thank you for presentation. I want to ask just one thing. Are you applying the same medical treatment to human, meanwhile the zebrafish? Because you can use some kind of medicine like effective materials inside the don't forget me flowers. You can use this medicine treat on human, so can you use it in the zebrafish too?
The potential therapies. Yes. Thank you for reminding. Zebrafish is perfect for drug screening. Yeah, they use it all the time for toxicity. In cell culture, you can test toxicity, but zebrafish is more effective. And for treatment, but the problem is, I think it treats zebrafish that it fails in human, because human have more like protective mechanisms, immune system wise.
Thank you.
Questions? Yoksa? Okay.
Question, but why the people with Alzheimer disease cannot forget that they have that disease?
Oh,
yeah,
perfect. Yeah. I think they forget it at the very end. And As far as I remember, it's like they don't, there is the semantic memory and episodic memory, and which I always forget, but like they never forget the semantic memory. I think it's when you remember Ankara is the capital, is semantic, right? So they never forget that, I think, I guess, as I remember, until the very end. Probably it's like that personal memory is affected only. And that's a very interesting topic, I think. And yeah, it's worth investigating.
Thank you for answering.
Thank you very much. I enrolled into Miri Besken, you know, from psychology department. I enrolled into her class this year. I only attended half of the classes, but I learned this from there. There is a question.
Hi. Well, sorry. Well, the most prominent cautions are like socializing or having a hobby for Alzheimer's, but I can't quite grasp the molecular level to that therapy. So how does that really work on a molecular level? Could you expand on that?
Yeah, socializing is critical, highly critical in everything we do. And it's been proven in many research. In Alzheimer, I don't know, but in neurogenesis, the making of new neurons, it continues into adulthood, and it even continues in the old people too. And it's mostly triggered by social interaction. For example, it is triggered by exercise. We know that. But if you do it outside, so if you do it outside because you run into people, you have a change of scenery, and you have lots of stimulus from outside. Probably social interaction gives us the most important stimuli for our brain. And it is, I don't know the molecular mechanism, obviously, it's a very good topic as well. But the depression, you know, depression stops neurogenesis. So brain just stops doing new neurons, or even the neurons start dying in the depression. And what was it? So in the zebrafish too, if you put an animal into depression, for example, it will cease to form new neurons. But further than that, it dies. So it is deadly for us, having no stimulus from outside or being close to the stimulus. So people commit suicide, but committing suicide is not an active, mostly, it's not an active decision. It just, the brain dies itself, like it's a inevitable consequence of depression. So it comes from that. So if the brain has already started degenerating and already started dying, you just increase its rate in isolation.
I understand, thank you.
But it's very interesting, yes.
Thanks. Hello. You said that we can't, we can't really test it because they can't really form childhood memories like in humans, so we can't really know if they forget their childhood memories, but can't we put them in a maze when they're very little, or I don't know, put them in a new cage and change it later, and maybe, and then put aggregated proteins in their brain, and maybe they'll remember their cage or the maze.
Umutcan, write that down. We will do that. Yes.
Keep me updated with the results.
Yes, thank you. The one problem would be it has to be a very strong stimulus for a fish, and it is hard to test that kind of strong stimulus. Like, for example, in our tests we use square shapes or round shapes. Even it is harder, but they learned that. But for the fish to remember it, we have to, I think, test it every day so that by repeated exposure, it will be known. Like in the square room, for example, there is food. So we have to do it repeatedly. Still, I'm not sure if they will remember that, but it can be tested, yes.
Thank you.
But then we will have really hard time with the reviewers, the journal reviewers, you know. They will immediately say, how do you know that the fish remembers that?
No.
So that's the question.
Okay, thank you.
We have to be convincing.
We thank our speaker for her speech. To make this thank you more meaningful, we donated UNICEF on behalf of each of our speakers.
We invite Zeynep Gülsoy to the stage, a member of the Bilkent Psychology Society organization team, to present the UNICEF donation certificate.